Hi, good morning, everybody. Um, so today we continuing talk about the glucose metabolism. Um, I guess last time we stopped here at this slide. So we already know that our body will need to maintain blood glucose level by a different kind of mechanism, right, in the fat and fasting conditions. And we also know in the fat condition, our body responds to increased blood glucose level after meal, like two levels of response, right? The first tier response would be increased blood glucose level can stimulate the insulin secretion by beta cell in pancreas. And the second tier response would be the basically respond to insulin action, right? Once insulin levels rises, and then the insulin will drive glucose go into the cells for metabolism, then will reduce the blood glucose level, right? And the very important transporter that we mentioned that is insulin-dependent transporter. Remember this transporter, GLUT4, right? And the two major tissues that produce high levels of GLUT4 and responsible for insulin-stimulated glucose uptake are skeletal muscle and adipose tissue. And now I wanted to get a little bit more detailed how GLUT4 responds to insulin and transport glucose into the cells. And this slide just represents the adipose cells and skeletal muscle cells. And see here, it's like nuclear and the cytosol and the whole cells, right? And once GLUT4 protein is synthesized, and majority of the GLUT4 proteins actually localize inside of cells. See the small circle represent the vesicles, membrane vesicles. So the GLUT4 molecules are located on the small vesicles, right? And these vesicles, basically majority of GLUT4 vesicles are inside of cells without insulin stimulation. In the basal condition, we call it basal condition, means that insulin levels are low, right? And once insulin levels increase in the circulation, in the blood, then insulin can stimulate the GLUT4 translocation from inside of cells to the cell surface, right? And then you see a lot of glucose. So GLUT4 localize on the cell surface membrane and be able to transport the glucose into the cells. So this is the mechanism, how insulin regulates the glucose transporter by GLUT4. Right? Basically, again, stimulate GLUT4 molecules translocation from inside of cell vesicles to the cell surface, and then be, allowed glucose being uptake by GLUT4. And during the fasting, you see not only GLUT4 protein levels lower compared to the fat condition, but also have a less GLUT4 molecules on the cell surface, right? So you wouldn't have a lot of glucose being uptake into the cell because of less GLUT4 molecules on the cell surface. So this is a mechanism, and try to remember how insulin, stimulate GLUT4 translocation and stimulate glucose uptake by particularly skeletal muscle and adipose cells. And in the disease condition, for example, insulin resistance or type 2 diabetes, the GLUT4 molecules levels of GLUT4 is decreased, and also you have a less GLUT4 translocation to the cell surface because the cell is resistant to insulin, right? You are not sensitive to insulin. So even you have a high insulin levels in a insulin-resistant patient, particularly, and, you have a higher insulin levels compared to normal patient, but this insulin does not function well, right? Not able to stimulate glucose translocation to the cell surface. So this is a very important reason in the insulin-resistant or type 2 diabetic patient that you have a lower glucose uptake by muscle and adipose tissue. So the patient have higher levels of circulating glucose, right? And the scap muscle, in addition to insulin stimulate glucose uptake, this mechanism, exercise itself, muscle contraction, can also stimulate GLU4 translocated to the cell surface, right? So this is through the different mechanism. You have a question? Right. Right. I mean, it's just not sensitive to insulin, but you have to have a very high levels of insulin to make the action. And so that's a good question. I'm not sure the percentage, but I think I mean, first of all, GLU4 level is lower. So I would say maybe I'll just guess, right? Right about 30-50%, the lower the GLU4 activities. And skeletal muscle, again, in addition, to insulin stimulation, skeletal muscle also responds to contraction, right? So both insulin and muscle fiber contraction can stimulate GLU4 translocation to the cell surface in the skeletal muscle, right? So skeletal muscle uptake glucose by two mechanisms: insulin stimulation and exercise, right? Muscle contraction. So if you guys probably already heard about this, for type 2 diabetic patients, we also encourage people to do exercise, right? So exercise is one of the mechanisms you can, promote the glucose uptake into the skeletal muscle and reduce the blood glucose levels. Okay, and here we wanted to compare the two different glucose transporters, the role in the glucose control, GLU2 and the GLU4. First of all, tissue distribution. These two glucose transporters are distributed in different tissues, right? Remember, which tissues express high levels of GLU2? Tissues, organs? Pancreas and the liver, right? So liver and pancreas are highly express the GLU2. So you have a high levels of GLU2 in liver and pancreas. And what about GLU4? We just talked about it, right? Skeletal muscle and adipose tissue. So the distribution are different. They're different. And the second question that the role in the body response to dietary glucose. So for example, GLU2 in the beta cell in pancreas and respond to the high glucose level after meal, right? And the role of the GLU2 in beta cells is mainly to uptake a glucose, right? And then push or stimulate insulin release, right? So the role of GLU2 in the beta cell is to metabolize glucose and then it triggers signaling pathway in the beta cells and promote insulin release. And GLU4 is mainly in the muscle and adipose tissue, respond to the insulin, right, not to glucose. Respond to insulin, and GLUT4 translocated to the cell surface and uptake glucose from the circulation and to reduce the blood glucose levels. And uptake of glucose in this tissue be metabolized. All right, so the third question. So what happens to glucose homeostasis or glucose control if GLUT2 and GLUT4 is defective? For example, the patient have a lower GLUT2 levels. Let's thinking about the GLUT2 first. So if a patient has the lower GLUT2 levels in the body, particularly in the beta cells, what happened to the blood glucose levels of this patient? Say loudly. Can anybody? Increase, right? And can you tell me the mechanism why? How? If they don't have enough GLUT2, why their blood glucose levels insulin? Through what kind of mechanism? Yes. Yes, excellent. Yeah. Thank you. So because you have a lower GLUT2 levels in the beta cells, you're not able to take enough glucose to stimulate enough insulin release, right? So you have a relatively lower insulin level compared to normal person. So then the lower insulin levels were not able to stimulate enough glucose, GLUT4 translocation, and glucose uptake into the muscle and adipose tissue, right? So that will have a lot of glucose being still remain in the circulation, particularly happening after a meal, right, in the fat condition. And what happened to the GLUT4, if a person have the GLUT4 levels, lower GLUT4 levels, basically like insulin-resistant type 2 diabetes, right? The patient usually have a lower levels of GLUT4 in adipose tissue. But yes. Yes. So again, similar like to the GLUT2 deficient patient or defective patient, the lower patient with lower GLUT4 levels also have a higher blood glucose, right? Because you have not able to take up enough glucose from circulation. So again, patients with defect either GLUT2, GLUT4, or GLUT2 or GLUT4 have the same consequence, right? Have a raised blood glucose level, but the mechanism is different. If I asking one more question, what about insulin level if you compare this two different patients, like GLUT2 deficient patient versus GLUT4 deficient patient? Insulin levels. See that again? Yeah, okay. Yeah, I know what you mean. So GLUT2 have a lower insulin level, right? And GLUT4 have a high insulin level. So, all right. So now I'm going to question. Oh, you mean type 1 diabetes? Type 1 diabetes basically is due to the defect of insulin secretion, right? I don't think they're related to GLUT2, but some other different mechanism. They can make insulin, the different mechanism. Thank you, that's a good question though. So now let's talk about the glucose metabolism in specific tissues. We remember we mentioned, different tissues, they metabolize glucose differently in different physiologic conditions, right? So now let's talk about liver. So liver is a central organ for glucose metabolism. Liver can do all five different glucose metabolic pathways, right? Glycolysis, glycogen synthesis, glycogen breakdown, gluconeogenesis, and the pentophosphate pathways, right? So 5 different metabolic pathways, glucose can all occur in the liver. Again, depend on the physiologic condition, which pathway be activated and which pathway be inactive, depend on the physiologic condition, right? So this represents the liver cells. So you can see how liver metabolize glucose in almost 5 different pathways, right? First of all, glucose can be uptake into the liver by GLU2, right? Remember, and then can go to the glycolytic pathways, and then TCA cycle in the mitochondria for complete oxidation, and in the fasting conditions, then liver try to save glucose not to be break down completely and save the 3 carbon skeleton, and so pyruvate can be converted lactate in the fasting conditions. And glucose in the fat condition, glucose also can be utilized to synthesize glycogen, right? So glycogen synthesis pathway. And during fasting, glycogen can be break down and to release glucose in the fasting condition, right? And glucose can also be metabolized through the pentophosphate pathways, right? And gluconeogenesis, and precursors like lactate, the pyruvate, and the most important precursors that can be used to make glucose and released into circulation during the fasting condition, right? So it's very important, a very active organ that metabolize glucose, liver. Again, just try to remember in which physiologic conditions that which pathway be activated, right? And the skeletal muscle. So skeletal muscle, the pathways, glucose can be metabolized completely through the glycolysis and completely oxidized into CO2 and water in the TCA cycle, mitochondria. And also glucose can be synthesized glycogen, right, in the skeletal muscle, stored in the skeletal muscle in the fat condition, or during the resting conditions. And glycogen can be also break down to glucose. And this and the difference is between liver and skeletal muscle, the glycogen breakdown in the muscle primarily just for utilize for energy, right, and not release glucose to raise the blood glucose levels. So glycogen breakdown mostly happen during the exercise, right? And muscle is also very important contributor to the gluconeogenesis by releasing the lactate. So lactate being generated from glucose metabolism, glycolysis pathway can be released into the circulation during the fasting condition. So this lactate will be very important and transported to the liver for gluconeogenesis, right, for making glucose during the fasting condition. So just try to be familiar with this sort of diagram, right, to thinking about what kind of metabolic pathways that that can occur in the muscle, in the liver, or other tissue cells. And this question I think I asked you before, so does skeletal muscle release glucose into blood? It's not, right? And the reason we are will not directly release glucose into circulation because muscle lacks the glucose 6-phosphatase, right? We will talk about this later again. But skeletal muscle actually contributed to the what am I trying to say, to the gluconeogenesis during fasting condition by providing the precursors, gluconeogenic precursors like lactate, we just talked about it, right? And also alanines, because here we're just talking about glucose, so we haven't mentioned the protein metabolism. So muscle is also a very important tissue that metabolizes proteins, right? So metabolize proteins, muscle can release alanines. So alanine is also very important precursors to glucose. So by providing lactate and alanine, and these are the two important precursors of gluconeogenesis. So indirectly contributed to blood glucose levels, right, by providing precursors gluconeogenesis. But again, not directly release glucose into the circulation. And brain, we know that is very, in terms of energy needs, and brain heavily rely on the glucose, right, for ATP production. And glucose can usually completely oxidize in the brain. Not much lactate produced, right? So in the brain, the glucose is completely oxidized to produce CO2 and water and produce a lot of ATPs, and the reducing equivalent, right, for ATP production. And red blood cells. Red blood cells is the cells that only utilize glucose for energy, right? Cannot use fatty acids, cannot use the protein for energy, only utilize glucose for energy. The reason you guys probably already know, right? What's missing? Mitochondria. So red blood cells do not have mitochondria. So that's the reason glucose only be able to metabolize through the glycolytic pathway and generate lactate, right? So pyruvate cannot be further completely oxidized in the TC cycle because no mitochondria, right, in the cells. Adipose tissue. So adipose tissue, the glucose metabolism in adipose tissue is a little bit different from the other tissues, right? So adipose tissue can do glycolysis and also complete oxidation to the further pyruvate, acetyl-CoA and TCA cycle to produce energies. And the glucose can also be metabolized, just stop at this stage, the glycerol 3-phosphate. Glycerol 3-phosphate is the backbone of the triglyceride, remember, right? So this is the important substrate for the synthesis of triglyceride. And some of the acetyl-CoA that initially metabolized from glucose can be used for synthesis of fatty acids, right? So again, here I just mentioned, we will talk about a lot later, in the lipid metabolism. So acetyl-CoA is very important central molecule as the substrate of fatty acid synthesis, right? So, but the fatty acids for triglyceride synthesis in adipose tissue mostly actually from circulation, right? And the conversion from acetyl-CoA to fatty acids, this pathway just contribute a little bit of fatty acids for triglyceride synthesis, right, in adipose tissue. But majority of, again, the fatty acids for synthesis of triglycerides are from circulation, and initially from the liver. So triglyceride synthesized, and this pathway is from glucose to triglyceride synthesis basically occurred in the fat condition, right? When adipose tissue, adipose cell takes a lot of glucose from the circulation and convert it to fatty acids, particularly for glycerol-3 phosphate, right? So glycerol-3 phosphate, most of the glycerol-3 phosphate for triglyceride synthesis is from glucose, right? It's from conversion, converted from glucose, right? And it is, again, mostly occurred in the fat condition. And during the fasting, triglyceride can be break down into free fatty acids and glycerol and released into the circulation and picked up by other tissues for energy, right? So this process, we will learn about it later, called lipolysis, right? So break down triglyceride, release fatty acids is the lipolysis process, and this process usually more active in during the fasting condition, right? So just try to remember in terms of a glucose metabolism in the adipose cells, particularly during the fat condition, glucose can be converted to glycerol phosphate to provide the substrate for triglyceride synthesis in the fat condition, right? And again, the fatty acids, majority of fatty acids actually from the circulation, the pickup by the adipose cells, and together with glycerol-3 phosphate for synthesis of triglyceride, right? Any questions about this process? Okay. All right, so now let's continue the glucose oxidation. So once the glucose take up the by cells and go to the glycolytic pathway and generate the pyruvate, right? And pyruvate will be transported into the mitochondria for complete oxidation. So, and now we just wanted to get more detailed the glycolytic pathways and TC cycle and complete oxidation, right, for glucose. Now let's talk about the first metabolic pathways of glucose, glycolysis. So glycolysis is the first step in the glucose utilization as energy. So any type of cells, once they pick up glucose, always can do the glycolysis first, right? So any type of cells can do glycolysis. And this is the main pathways for metabolism, not just the glucose, but also the fructose, galactose, and other carbohydrates, right? And again, some cell types actually depend only on the glycolysis for energies, like we mentioned, the red blood cells, right? So now we get into like a little bit the detailed enzymatic pathways in the glycolysis, right? So glycolysis, we can divide it into the two stages, first stage and second stage. So this slide just showing the first stage. So the first stage basically is the consume ATPs, right? Not generating ATPs. So first stage, consume ATPs, and start with the glucose, and glucose can be metabolized or break down into the 2,3 carbon molecules, right? Glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. And these 2,3 carbon molecules can be interconverted, right? So remember the glucose is a 6 carbon molecules, right? And then in the first stage, the 6 carbon glucose molecules being break down into the 3 carbon molecules. And then these 3 carbon molecules then enter the second stage and further metabolize, right? So what you need to remember is that the rate-limiting reactions. So first reactions that glucose being phosphorylated to glucose 6-phosphate is the rate-limiting reactions, meaning that this reactions is controlled, right? And this reaction is catalyzed by the enzyme called glucose kinase or hexokinase. Why we give the two different enzyme names here is actually depend on the tissues. Which, for example, in the liver, the glucokinase is more highly abundant, and hexokinase is more abundant in the skeletal muscle. So in the different tissue, they give a different name. I don't know why, but they catalyze the same reactions. All right, there are two different enzymes in different tissues catalyze the same reactions for the glucose phosphorylation. So again, this is the first rate-limiting reactions and consume the 1 ATP. And the second rate-limiting reaction is the conversion of fructose 6-phosphate to fructose 1,6-biphosphate. And enzyme for this reaction is phosphofructose kinase, right? And this reaction also utilizes 1 ATP molecules, right? So in the first stage, you try to remember these 2 key reactions or rate-limiting reactions and the key enzymes, right, to catalyze this reaction. So try to remember what is substrate, what is the product of these 2 key reactions, right, and enzymes. And again, this is ATP consuming stage. Right, so the second stage, so start with the glyceraldehyde 3-phosphate, which break down from the glucose, right? So you generate actually 2 these 3 carbon molecules, so enter the second stage. So second stage is the ATP generating process, right? So you generate ATPs during this second stage, and you generate ATPs directly from the substrate phosphorylation levels. So you see two reactions. They're producing the ATPs directly from the substrate levels phosphorylation, right? And in addition to the substrate levels phosphorylation mechanism, you generate direct ATPs, and you also produce the reducing equivalent, NADH, right? NADH is very important, that can provide energy. So NADH basically will have to transport electron from its molecules to the electron transport chain in the mitochondria for oxidative phosphorylation and generate the ATPs there, right? So in other words, that NADH has to go into the mitochondria to the electron transport chain and produce ATP there, right? So, and in the second stage, you also try to remember the key reaction or limiting reactions, which is catalyzed by pyruvate kinase. So this kinase catalyzed conversion of phosphoenolpyruvate to pyruvate, right? Try to remember because in the second stage, two glyceraldehyde 3-phosphate enter this stage, right? Remember? So you will generate actually 2,8-pyruvate from the second stage, from the initial 1 molecules of glucose. Does that make sense? Right? Okay. So and what about ATP production? 4, yes. 4 ATPs actually produced, right, from the substrate phosphorylation levels, and 2 NADH, right, molecules produced from this stage. And the other important thing that I wanted to point out is that the final product of the glycolysis can be different depend on the metabolic conditions. For example, in the aerobic metabolism conditions, that means that you need oxygen and cell has a mitochondria, and the pyruvate can be further metabolized, can enter mitochondria and for complete oxidation through the TCA cycles in mitochondria, right? So in the aerobic conditions, the final product of glycolysis is pyruvate, right? But under the anaerobic conditions, for example, in the red blood cells, right, they don't have a mitochondria, so they cannot do the aerobic metabolism. So what happened is here, the pyruvate is converted to lactate in the anaerobic conditions in red blood cells or in the skeletal muscle. Skeletal muscle only during the very fast the exercise, for example, 100 meters sprinting, and muscle cells only does glycolysis to metabolize glucose and produce ATPs. But under the anaerobic conditions, so pyruvate is converted to lactate, and the most important thing is that this reaction requires the NADH, right? So the NADH produced, at these first reactions during the second stage can be used for the conversion of pyruvate to lactate, right? So in the anaerobic conditions, the ATP production can be different through the glycolysis pathway, can be different from that in the aerobic condition, right? So that means that in the anaerobic condition, the NADH produced here being utilized here. So that means you have a net 0 NADH produced, right, in the glycolysis pathway under the anaerobic conditions, right? Does that make sense? Okay. So now let's do some kind of exercise to try to emphasize this understanding, the ATP production, right? So let's do the aerobic conditions. So how many ATPs are produced in aerobic condition during glycolysis from 1 molecule of a glucose? Right, the end products should be pyruvate, right? And how many ATPs? are produced 4 2. Remember, why is 2? You utilize 2, right, in the first stage. So you produce the 4 in the second stage, but actually utilize 2 in the first stage. So that's why the net production ATP is 2. And what about NADH? 2, yes, correct, 2. And how many ATPs you will expect to be generated from 2 NADH through the electron transport chain? 6? Okay. 4 and 6. Why 4? Why not 6? Sometimes 4, sometimes 6. Depend on the different tissue cells. We're going to talk about it later, because of remember, the NADH produced in the cytosol. Again, I forgot to mention, the glycolysis occurs in the cytosol, right? So, an electron transport chain is located in the mitochondria. So NADH produced in the cytosol from glycolysis need to be transported into the mitochondria, and because it's not membrane permeable, so not freely, it's not able to freely enter the mitochondria. So it requires a transporter or shuttle system, and depend on which shuttle system that transports NADH into the mitochondria, then determine how many ATPs are produced in the mitochondria. Right? We will get there later. All right? Let's just try to remember, this. Okay, so in the aerobic conditions, we produce like 4 to 6 plus 2, right? The ATPs in cells under anaerobic condition. So actually total 6 to 8, right? ATPs in aerobic conditions. Okay, what about under the anaerobic conditions? How many ATPs produced? Should be same, right? Like we did. For ATP, substrate phosphorylation level should be same, right? 2. 4 produced in second stage and 2 used in the first stage, so net will be 2. And how many NADH? 0, yes. So you produce 2 ATPs under anaerobic conditions. For example, in red blood cells, right? You will expect just 2 ATPs produced through the glycolysis pathways. And again, final product is the lactate. So if you compare to the brain is always be able to completely oxidize glucose, right? So we'll consider the aerobic conditions. So brain will be 1 molecule of glucose is oxidized in brain cells should produce 6 to 8, right, ATPs. And 1 molecule of glucose is oxidized in red blood cells only produce 2 ATPs. Right? So under different metabolic conditions. Okay, so I'm not going to go through this and just try to remember, this 4 ATPs and produce in which states, which reactions, and 2 ATPs consumed, right, in the first stage. All right. So let's just finish this slide and then we take a break. So what happened to the pyruvate that is formed from the glycolysis? Assume that the cells had the mitochondria under aerobic conditions, right? So pyruvate, once it's metabolized from glucose, transported into the mitochondria and go further being break down to the acetyl-CoA. This process called hydrogen by pyruvate dehydrogenase is a very important process, right? So, and once the acetyl-CoA is generated from glucose metabolism and enter the TCA cycles for complete oxidation, produce CO2 and water, right? And pyruvate, this is the one important pathway for complete oxidation, right? And pyruvate can also be converted to lactate. Again, this under the anaerobic conditions. And pyruvate, if further metabolized to acetyl-CoA enter TCA cycles, and acetyl-CoA first reactions can be together with oxaloacetate be condensed to citrate, right, in the TCA cycles. Citrate is very important molecules can transport out of mitochondria back to the cytosol and converted back to acetyl-CoA. Again, I mentioned acetyl-CoA is the substrate of fatty acid synthesis, right? So acetyl-CoA can be used to synthesize fatty acids, palmitate here as an example, right? And this again process occur in the fat condition when you, and particularly in the liver, right? When you have a lot of glucose, you get from diet, and glucose can be converted to fat. This is the process, right? So the conversion of glucose or carbohydrate to the fat, particularly in the liver, happening in the liver, right? And again, still in the liver, the acetyl-CoA, which converted to pyruvate, can also be utilized for the synthesis of ketone bodies, right? And this ketone body synthesis process, again, occurs in the fasting condition, right? And I mean, just wanted to emphasize that again. The ketone body synthesis occur in the fasting condition only in the liver. Only liver is able to synthesize ketone bodies, right? Okay. Yeah, let's take a 10 minutes break. Okay. All right. Now let's talk about the other sugars, how they metabolize through the glycolytic pathway. And here I just wanted to talk about the fructose, right? Um, and again, fructose compared to glucose in our diet is less abundant, right? Unless you eat a lot of, the fructose. For example, corn syrup. If we consume too much corn syrup, then you actually get a lot of fructose. So fructose is, I think it has kind of bad reputation, right? Kind of connected to obesity and fatty liver disease. And so let's just, take a look at how fructose is metabolized, why we eat too much fructose and have some, the metabolic problems, the obesity and fatty liver disease. So if you compare to glucose, and fructose is metabolized by a different enzyme, particularly for the first step reaction. For glucose, the first reaction, key rate-limiting reaction is the glucokinase or hexokinase, right? And converted glucose to glucose 6-phosphate, and then enter this glycolytic pathway by generating the 3-carbon molecules, right? This first stage for the glycolysis. And fructose goes to different pathways to generate 3 carbons, the same 3 carbon molecules. Again, the fructose kinase is a very important enzyme that metabolizes fructose. And also aldolase B is also important enzyme, right, in the fructose glycolytic pathways. And the second thing that you want to know is the fructose primarily is metabolized in the liver. And the reason why fructose is metabolized primarily in the liver, because these 2 enzymes are highly abundant in the liver compared to the skeletal muscle, right? In the liver, you have a high levels of fructose kinase and aldolase B. And in the skeletal muscle, you only have these 2 enzymes, and that's very highly, high levels in the skeletal muscle. So basically, metabolize glucose mostly in the skeletal muscle, right? So, and the other thing is the fructose, not like the glucose, fructose does not stimulate insulin secretion, right? So why is this important? I will explain this in the next slide, right? So just go through the pathways of fructose glycolysis. So fructose, first of all, is converted to fructose 1-phosphate by fructose kinase. And then the fructose 1-phosphate is break down to the 2 3-carbon molecules and by the aldolase B, right? So aldolase B is break down fructose 1-phosphate and to the 2 3-carbon molecules. And these 2 3-carbon molecules can all be converted to glyceraldehyde 3-phosphate, right? And then go to the rest of the glycolytic pathway, particularly the second stage of glycolysis, right? So the difference is only in the first stage, right, between fructose and glucose, and by the enzyme, and it's also different in the first stage. And because if you remember this reactions, conversion of fructose 6-phosphate to fructose 1,6-phosphate by enzyme called phosphofructose 1 kinase. And this is the rate-limiting reaction, right? Rate-limiting reaction means once you have a lot of glucose be metabolized and you have a lot of fructose 1,6-biphosphate generated, and this substrate is strong inhibitors of phosphofructose 1 kinase, right? So once you generate a lot of fructose 1,6-biphosphate, and this substrate in a negative feedback mechanism to inhibit the phosphofructose 1 kinase. So then to slow down this glycolytic pathways, right? So this is the mechanism for how this reaction is considered rate-limiting reaction. However, the fructose metabolism actually bypass this rate-limiting reaction, right, in the first stage. So that means that you can have a lot of glycoaldehyde 3-phosphate generated from fructose metabolism, right? So then you have a more glycoaldehyde 3-phosphate enter the second stage and to generate more pyruvate and the product of a glycolysis, right? Does that make sense? Any question about this? So, I mean, the reason is, the fructose glycolysis, bypass this rate-limiting reactions. So compared to glucose and fructose, you have generated more glycoaldehyde 3-phosphate, right? Does that make sense? Right? And then you have more pyruvate generated. And pyruvate, we talked about, I think, the couple slides earlier, that pyruvate can be converted to acetyl-CoA and then to TC cycle, citrate, and fatty acid synthesis, right? So if you have a lot of pyruvate being produced from fructose metabolism, then you have a lot of, substrate of fatty acid synthesis. So a lot of fructose can be metabolized and then converted to fat. So that is one mechanism for why we, consume too much fructose and can be converted to fat in the liver. Any questions? Right? Is that clear? Okay. So, and this, again, here I just simplify a little bit, right? The pathways. So, and because fructose does not stimulate insulin release, the insulin release is important in the fat condition that can convert it glucose to the glycogen and store in the liver, right? So you have a lot of glucose and absorb it from the diet, but you're also able to stimulate the glucose is able to stimulate insulin. So the insulin, one of the important function of insulin is to stimulate glycogen synthesis from glucose, right? So it's okay. This sort of the other mechanism that you can try to reduce the burden of the glucose, right? So you store the glucose in the glycogen, and this process is promoted by insulin. So this okay, but the fructose does not stimulate insulin. So this process will not happen, right, in the liver when you consume a lot of fructose. So fructose, will all fructose be metabolized to this pathway, right? So, and produce pyruvate in the second stage of glycolysis. And pyruvate, again, I mentioned, can be further metabolized to acetyl-CoA and citrate and for fatty acid synthesis. So just by comparing fructose to glucose, and fructose is more easy to be converted to fatty acids, right? The two reasons, one is I mentioned, the fructose metabolize, bypass the rate-limiting reactions. And so that means that fructose can be metabolized without control, right? So you will have more, glucose being converted to pyruvate. And the second reason is fructose does not stimulate insulin release, so the glycogen synthesis pathway will be also blocked. So you have this fructose 6-phosphate that generated from fructose cannot go this pathway for glycogen synthesis, right? So all be pushed to this pathway for pyruvate synthesis and converted to the fatty acids. So this is this sort of the main mechanism. So why if you have high consumption of fructose causing a lot of problems, particularly related to lipogenesis in the liver and fatty liver and obesity, right? That clear? Any questions about this? Right? Okay. Yes. Mm-hmm. Right. Yeah, this pathway. Oh, this will happen, but not like a lot, because this cannot be, cannot be further converted to glycogen, right? This pathway will not happen much without, because it doesn't stimulate insulin, right? So, so the majority of fructose will go this pathway, right, for pyruvate production. All right. And the other disease that related to fructose metabolism is called fructose intolerance. And fructose intolerance is hereditary disease. And the reason of the fructose intolerance is the patient actually lacks the adelase B, I would say, right? More precisely, adelase B deficiency. And the problem that this patient will have is the patient will experience the hyperglycemia. I mean, they cannot maintain their blood glucose level, particularly during fasting, right? And let's go to the detail why they cannot. The patient, had this blood glucose control problem. As you see here, we already mentioned, right? Fructose is metabolized fructose 1-phosphate by fructokinase, and then fructose 1-phosphate is break down into the 2,3 carbon molecules by adelase B, right? So once the patient does not have this adelase B, then fructose 1-phosphate cannot be break down, right? And then accumulated. So fructose 1-phosphate is strong inhibitors of glycogen breakdown, right? So because of lack of this enzyme, so you have fructose 1-phosphate accumulated, and this will strongly inhibits the glycogen breakdown and to release glucose. So during the fasting, we need to break down glycogen, right, to raise blood glucose level. And if we don't have this enzyme, and then the glycogen cannot be broken down to release glucose. All right. Any question about this fructose metabolism? Right. Now let's move on the rest of part of the glucose metabolism that involve the mitochondria, right? Complete oxidation in the mitochondria. So first of all, we wanted to get familiar with the mitochondrial structure. You probably all know this, right? Mitochondria has a double membrane and inner membrane, outer membrane, and matrix. And the important thing that I want to point out that all the TCA cycles, enzymes, actually are in the mitochondria. And the respiratory chain, or ATPase, are also localized in the inner mitochondrial membrane, right? And PDH, which is the pyruvate dehydrogenase complex, is also in the mitochondria. So all these enzymes are localized in the mitochondria. So the mitochondria is very important. The oxidative metabolism for all the three nutrients, glucose, amino acids, and fatty acids, right? And pyruvate dehydrogenase complex is the three enzymes that form the complex, right? I don't want to go to the detail, but just let you know that it's a membrane as an enzymatic complex, right? Contains the three different sets of enzymes and catalyze this is a whole reactions. They catalyze the decarboxylation of a pyruvate, so you remove the one carboxyl group and generate CO2, and this is a product of this reaction, the acetyl-CoA. Acetyl-CoA is very important, right? Again, the central molecules, and this reaction occurs in the mitochondria and is also very important for the energy metabolism. And other thing is this reaction also generates 1-NADH molecules, right? And this enzyme activities is also controlled by several things, and two important things is that its product, for example, NADH and acetyl-CoA are the product of pyruvate dehydrogenase catalyzed reaction, right? And once you have a lot of, the acetyl-CoA or NADH is produced, then these two molecules can serve as sort of negative inhibitors, right, to block or to reduce the pyruvate dehydrogenase activity, to not allow the too much energy being produced from the pyruvate, right? So again, these are the two important product inhibitors or substrate inhibitors of pyruvate dehydrogenase. And again, this actually just gives you a brief overview. The acetyl-CoA not only be able to generate it from glucose metabolism, but also from fatty acids and amino acids metabolism, right? So again, acetyl-CoA is essential molecules in terms of the energy metabolism from three macronutrients. Amino acids, glucose, and fatty acid metabolism can all generate the acetyl-CoA, and acetyl-CoA can be completely oxidized, so it's an important molecule that enters TCA cycles, right, for complete oxidation. And acetyl-CoA is also important substrate for synthesis of fatty acids, ketone bodies, and cholesterol, right? So see how important acetyl-CoA molecules, so try to remember to connect all, the three macronutrient metabolism, right? And it also does the, an important role in regulating the conversion of the one macronutrient to the another macronutrient. For example, convert glucose or amino acids to the fatty acids, right? TCA cycles. I think this is very nice, actually, diagram to show you, you can connect all three macronutrients and in terms of energy metabolism. Acetyl-CoA, again, is essential molecules, right, produced from metabolism of carbohydrate, protein, lipids. And this is the key molecule that can enter TCA cycles, right, and to produce ATPs and also reducing equivalent like NADH or FADH, and so reduced from the TCA cycle, produced from TCA cycles. And then the reducing equivalent NADH or FADH eventually will need to go into the electron transport chain for oxidative phosphorylation and for eventually for producing the ATPs in the electron transport chain, right? And all this process, the TCA cycles and electron transport chain are in the mitochondria, right? And TCA cycles, I don't think you guys need to remember all the reactions, right? But just when you look at the TCA cycle and you can get some sense, right? How TCA cycles connect the glucose, fatty acids, amino acids metabolism, right? And what is the main function of the TCA cycles? So first, the function of TCA cycle is to produce energy, right? To oxidize nutrients, complete oxidized nutrients, produce ATPs and NADH, FADH. This is one of the important functions. And the second function of TCA cycles is to involve in the gluconeogenesis, right? Because remember well, actually we haven't talked about it yet, right? The gluconeogenesis basically is converted non-sugar precursors that including amino acids, right? And glycerol lactate to the glucose. So lots of amino acids can serve as a precursor for gluconeogenesis, and the TCA cycle is very important pathway for gluconeogenesis from amino acids, right? So the second function of TCA cycles is to be involved in the gluconeogenesis, right? And also, fatty acids, right? Can be the citrate is converted from acetyl-CoA, can be used for fatty acid synthesis, right? So TCA cycle is also part of the lipogenesis pathways. And in terms of the ATP productions, you see that one ATP is produced at the Sept can directly produce from the TCA cycles, right? And then you also can see three NADH molecules produced per one cycles of TCA and one FADH molecules produced, right? And also, when you calculate the ATPs, you probably also wanted to thinking about that the one acetyl-CoA enters the TC cycles, and if we oxidize one glucose, then we usually generate two acetyl-CoA molecules, right? So when you calculate ATPs, you have to double the ATP numbers, right, per TC cycles from the one glucose oxidation, right? So ATP production from complete oxidation of one molecule of glucose. So you need to thinking about the glucose oxidation start from glycolysis, right? And then to the pyruvate. And pyruvate converted to acetyl-CoA, and acetyl-CoA enter TC cycle, right? So thinking all the whole process and try to calculate, the ATPs. I think the ATP, how many ATPs, the number of ATP production is not really important. I don't really care. I mean, but just I think this, if you through that calculation, just help you understand the whole, the metabolic process, right? How glucose is metabolized for energy production. So eventually, one glucose molecules, if it's complete oxidized through glycolysis, TC cycles, and generate 36 to 38 ATPs, right? So you maybe go back and to calculate how this number is, comes out, okay? All right, I think we finished this unit. Okay, now, I think we talk about this, right? The anaerobic conditions, and we usually occur in the cytosol, and end product is lactate, right, and produce only 2 ATPs from one glucose, and glycolysis plus TC cycle that for complete oxidation of glucose, right? And the glycolysis pathway from glucose to 2 molecules of pyruvate that occur in the cytosol, and then pyruvate be transported into the mitochondria, and the rest of the process all occur in the mitochondria. Pyruvate dehydrogenase converts pyruvate to acetyl-CoA, and acetyl-CoA enter TC cycle and for producing ATPs, right? And NAD, FADH enter the oxidative phosphorylation process for the actual ATP production, right? And now I wanted to talk about a little bit detail the respiration chain or oxidative phosphorylation. If you guys still remember that respiratory chain actually consists of 4 complexes, right? Complex 1, 2, 3, and 4, and each complex has the multiple enzymes, right? Again, it's also the multiple enzymatic complex, and actually lots of, and each complex does a different functions, right? For example, complex 1 basically serves as NADH dehydrogenase activities, and complex 2 has a succinate dehydrogenase activities, and 3 the chromosome BC1 complex, and 4 is chromosome C oxidase. But you don't have to remember each enzymes, but just thinking this is a complex a lot of enzymes, right? And involve the oxidate and oxidation reduction reactions, right? So, and complex 1, 3, and 4 are proton pump. This is very important, right? Because the whole process, through the electron transport, you generate the proton gradient across the inner membrane, mitochondrial membrane. So this proton gradient contains energy, so this energy can be transferred to ADP molecules and to generate ATP, all right? So proton gradient is very important. So only complex 1, 3, and 4 can function as a proton pump, so can pump a proton out of mitochondria, so and generating the gradient, right, proton gradient. So here's a diagram that I think it will have a better idea. First of all, you can the location of the complex 1, 3, and 4 localized in the inner mitochondrial membrane, right? And complex 2 is localized in the matrix, mitochondrial matrix. So this probably is the reason why this 3 complex can serve the proton pump, right? Because across the membrane, so can pump up proton out of inner mitochondrial membrane and generating the proton gradient. And complex 1 is in because of its location, so does not have any function as the proton pump, right? So that means so any electron that transported through complex 1, 3, and 4, it can be able to generate energy and ATPs. If transfer proton to the complex 2, will not able to generate proton gradient and energy, right? So, so for example, NADH transfers proton to complex 1 and then 3 and 4. So NADH, so that's the reason why NADH is able to generate 3 ATPs, right? So each complex creates the protons at enough for 1 ATP production, right, energy. So NADH transport, through these 3 protons, generate 3 gradient. So that they be able to produce ATPs, right? So proton gradient eventually is being, proton being transported back into the matrix and release energy and transfer energy to ADP and to ADP and generating ATP, right? And FADH, because FADH transfers proton to complex 2 and then bypass complex 1 and then complex 3 and 4. So FADH is only able to generate 2 ATPs, right? That's the reason. All right, now here I wanted to explain that we talk about during the glycolysis. Sometimes NADH be able to generate the 3 ATPs and sometimes 2 ATPs, right? Remember, and so here's the reason. I'm going to explain the 2 transporter shuttle system that help the transporter of NADH from cytosol to mitochondria for ATP production. And first shuttle system is called glycerol 3-phosphate shuttle. So NADH is generated through glycolysis in the cytosol. Again, itself is not membrane permeable, so it's not able to transport through the mitochondrial membrane itself. So it requires the shuttle system to help its transport into the mitochondria. So first of all, NADH gives the proton to these molecules and then convert it to the glycerol 3-phosphate, right? And glycerol 3-phosphate that passes proton or electron from NADH to the FAD, right? And then generate FADH. So glycerol 3-phosphate is able to pass through the outer membrane, right? And then passes the proton or electron from NADH to FADH, and then generating FADH. Because FADH only produces 2 ATPs, right? FADH transport electron to complex 2, and then 3 and 4, so only produce 2 ATPs, right? So if NADH generated from glycolysis is transported through glycerol 3-phosphate shuttle system, it's only be able to produce 2 ATPs. Does that make sense? Right? Because this FADH is the actual reducing equivalent that eventually then directly enter the electron transport chain for ATP production, right? Does that make sense? Any questions? Right? Okay, so the other system called a Mealy aspartate shuttle. Again, if NADH proton is transported into the mitochondria by Mealy, it's different. So Mealy is able to pass the electron from NADH and to the NAD inside of mitochondria, right? And so NADH is a reducing equivalent that directly, goes to the electron transport chain for ATP production. So through this transport shuttle system, this NADH produced in the cytosol is able to produce the 3 ATPs, right? Is that clear? So again, why NADH produced from glycolysis can produce, sometimes produce 2, sometimes produce 3, depend on the shuttle system. And shuttle system is not, the 2 shuttle system not always present in the one type of cells. So some type of cells only maybe have a glycerol 3-phosphate shuttle, and other cell types have Mealy aspartate shuttle that do not have much glycerol 3-phosphate shuttle, right? So depend on type of cells and if they have, which shuttle system they have, and then that determines how many ATPs are able to produce from the glycolysis generated NADH, all right? And in the TCA cycles, the NADH or FADH produced from the TCA cycles will always produce NADH that are produced in the TCA cycles always produce the 3 ATPs. Why? Because they're already inside of mitochondria, right? They don't need the transport system. So they can directly go to the electron transport chain and produce the 3 ATPs. right? Okay, so now let's talk a little bit about the gluconeogenesis. So gluconeogenesis basically is the biosynthesis of glucose from the non-carbohydrate precursors. And the main function of glucose, why our body need to do gluconeogenesis? Why we need to do gluconeogenesis? What is main function? Why we need to do this? To produce glucose and try to maintain glucose levels, right? When we do need to maintain glucose level, add glucose to the circulating. During fasting, right? So under fasting conditions, gluconeogenesis are very active, and the main purpose of gluconeogenesis is to produce glucose and to maintain or to raise the blood glucose level. So it occurs primarily in the liver and kidney. Liver and kidney are the main tissues that do the gluconeogenesis, right? And it primarily, in terms of intracellular compartment, so gluconeogenesis can occur in both places, in mitochondria and the cytosol. So some reactions in the mitochondria and then the other reactions in the cytosol, right? So, and it's not exactly the reversal of glycolysis, right? And the common precursors of gluconeogenesis that you probably try to remember: pyruvate, lactate, glycerol, and propionate. And also some amino acids will also be able to serve as precursors for gluconeogenesis. This type of amino acids we call the gluconeogenic amino acids, mean that they are able to be used for glucose synthesis or production, right? For example, alanine, serine, and glycine, any amino acids that can be converted to pyruvate or oxaloacetate or alpha-ketoglutarate. These amino acids are all able to use for glucose synthesis, right? And again, I mentioned that the gluconeogenesis is not the simply reversal of the glycolysis. The reason, because we remember we have three rate-limiting reactions, right, in the glycolysis, and this reactions that catalyzed by these three key enzymes. Just take one reaction as an example. For example, glucose can only be converted by the glucose 6-phosphate by hexokinase. So hexokinase is only be able to catalyze the conversion of glucose to the glucose 6-phosphate. And hexokinase is not able to convert glucose 6-phosphate back to glucose, right? So that means we need to have a different enzymes or reactions to convert the glucose 6-phosphate back to glucose. And similar situation for the other two limit rating reaction. Again, we need a different set of enzymes to convert it, to does the conversion, right? And so we have sort of three bypass reactions in the gluconeogenesis to catalyze the conversion of all these three reactions, right? And here are the three bypass reaction in gluconeogenesis, right? So start from this, pyruvate, which is the end product of a glycolysis, right? and convert it back to the phosphoenolpyruvate requires the two enzymes, pyruvate carboxylase and phosphoenolpyruvate carboxykinase. And these two enzymes are considered key reactions in the gluconeogenesis, right? And the second key bypass reaction is the catalyzed by fructose 1,6 biphosphatase, right? And this enzyme converts fructose 1,6 biphosphate back to fructose 6-phosphate. And this is also the key enzyme in the gluconeogenesis. And last one is the glucose 6-phosphatase that converts glucose 6-phosphate back to glucose. So remember, I mentioned this enzyme a couple times before. The muscle lacks this enzyme, right? So muscle cannot convert glucose 6-phosphate to free glucose. So that's why muscle is not able to release glucose from the glycogen breakdown. And glucose 6-phosphate is not only generated from glycolysis, but also from glycogen breakdown, right? So remember, these are the three key reactions in the gluconeogenesis, right? That's the conversion of the glycolysis, the key reaction in the glycolysis, right? And this just gives you the overview of the gluconeogenesis. Again, some part of the reactions in the, occurred in the mitochondria, and the rest of the reactions in the cytosol. And here we just give you example starting molecules for gluconeogenesis is a pyruvate. So how pyruvate being used for synthesizing glucose, right? Again, the two reactions in the mitochondria, and the other two reactions in the cytosol, right? And the other important thing is that depend on the substrate. The pyruvate is almost going to complete all the reactions. And if substrate is amino acids, and the process could be a little bit different. For example, the glutamate, they actually bypass the first reactions, only have one reactions in the mitochondria. And if the precursor is the glycerol, and you only see the two bypass reaction in the cytosol, and that's not required reaction in the mitochondria, right? Just depend on the substrate precursors, which precursors are used for gluconeogenesis. The pathway could be different. All right, let's just finish this, right? I think we have about 5 minutes, right? Okay, so let's say that if we use the pyruvate as the precursor, so first of all, pyruvate be transported into the mitochondria, and then this is the one reaction that pyruvate converted to oxaloacetate by this enzyme pyruvate carboxylase, and then the oxaloacetate is converted to phosphoenolpyruvate (PEP), right? And this is second key reaction in the mitochondria called PEPCK, or pyruvate phosphoenolpyruvate carboxylase, right? So two key reactions in the mitochondria that converts the pyruvate to PEP, and PEP then transported out of the mitochondria to the cytosol, right, and to complete the rest of the reaction. So this is the two reactions in the mitochondria if pyruvate used as the precursor of gluconeogenesis, right? Okay, so let's stop here for today. Okay. And we have a quiz next Tuesday, right? So I'm going to post the study guides for you guys, hopefully today, latest tomorrow, right?